welcome to Intro 101. My name is Chris, and I am your neuroscience and psychology graduate student assistant. I'm here to assist you with the related science basics that you interact with on a daily basis. My goal for doing this is to help you start or continue to build a foundation in human biology and physiology that can better help you engage with the information you consume from your favorite health and wellness educators and influencers. Much like the way a graduate assistant helps prepare intro students to better engage with lectures delivered by their professors. Or maybe you're here today because you just like to know what's lurking in the depths of your brain juice. Either way, stick with me for the next bunch of minutes and I'll do my very best to explain what's living in there and not at all rent-free. These darlings we're looking at today are incredibly hard workers. Today's episode is Intro to Nervous System Cell Physiology 101, Part 2. It is entirely devoted to the hardworking and underrated glial cells. For each type of glial cell, we'll go over basic cell structure, anatomy, and function, including how they not only support neurons and synaptic transmission, but also have all kinds of their own business independent of neuron admin. We'll also look at the blood-brain barrier as it works in cahoots with some glial cells, and we'll also cover some of the different ways glial cells are implicated in neurological disease states. I explained in the last episode how mighty the neuron is. Well, glia cells are no wimps either. They also have communication skills, plus they are strategic and highly active even when the rest of you is asleep. They have their own magic, and it took some brave and curious and creative souls to take a chance and study these underdogs, and I'm so glad they did, because you may be surprised at just how important they are to your daily existence. I'll introduce you to some of these inspiring neurobiologists and neuroscientists and try to give justice to their incredible contributions to unfolding the mystery of glial cells. And in the homework section of the show notes, I'll introduce you to their scientific progeny who carry on their legacy today, and you can read all the ways they have added to our growing understanding of these intriguing cells. Before we start, I'd like to respectfully acknowledge that Intro 101 is recorded on the unceded and ancestral territories of the Kwantlen, Katzie, Matsqui, and Semiahmoo First Nations. Additionally, we here at Intro 101 believe wholeheartedly that science is for everybody. In fact, the richness, complexity, and strength of scientific research is astronomically improved by the inclusion of many varied voices. To ensure everybody has access to education, research, resources, and representation across scientific fields the world over, I will be sharing organizations in the show notes that provide access to STEM education and research resources, and ask that you check them out and support them in any way that you can. You can donate, share their work on your social media networks, or maybe volunteer your time to help them. If you already advocate and generate opportunities to provide equitable access to STEM education and resources within your own communities, or support other STEM education organizations not listed here, please share them in the comments below if you'd like others to contribute as they are able. All right, welcome to class. Thanks for being here. Let's crack on with Intro to Nervous System Cell Physiology 101 Part 2, Glial Cells. First, I'd like to mention, if you've just arrived at this channel through this episode, this series is an episodic integration of information that is building a bigger picture of the nervous system, its components, physiology, and its collaboration with other body systems to keep your body in homeostasis. If you're new to homeostasis and or the nervous system, or a little rusty on how it works, it may benefit you to view the previous episodes in this series first. A general understanding on what homeostasis is, nervous system function, general cell physiology, and neuron function and physiology will help make sense of this episode, all of which are covered in the first 4 episodes of this series. Okay, glia. Are you ready? What are these mysterious cells? Well, first they live in the central and peripheral nervous systems and have many, many jobs. But when they were first discovered, they were considered the glue that kept neurons in their place. In fact, the word glia is often attributed to being derived from the Greek word for glue. But when I searched the etymology of glia, the Greek word glia or glios also means mucus, slime, or crap. So when German pathologist Rudolf Virchow first named them, I don't think he thought much of them. They were first identified in the 19th century by Virchow and a number of other scientists, including Santiago Ramon y Cajal, whom you'll recall from previous episodes. Over the next century, different types of glial cells would be identified, but glia mostly remained thought of as brain mucus until only just a handful of decades ago. But as we've grown to understand them a little better, it's been pretty well established that, yes, they do act as support crew for neurons, but in more recent years, we're learning that they're so much more than neuron staff. Some of the ways they do support neurons is by, yes, holding them in place. They also regulate and supply nutrients to neurons so they can make the ATP they need to burn 4.7 billion ATP molecules per second per neuron in a resting state. See the previous episode for context. But right there, that's a huge job. That's Costco-sized nutrient demands. Glial cells also insulate neurons from each other and keep electrical signals on track. They are involved in multiple stages of neuron life, having roles in a neuron's development, its growth, its repair when injured. They even clean up dead neurons. They gobble up degraded neurotransmitter bits that can't be recycled, and they assist in other aspects of synaptic communication. Outside of neuron-specific support, some glia are involved in creating cerebral spinal fluid. Some clean up plaques and pathogens. Some are involved in the maintenance of the blood-brain barrier as well as the lymphatic pathway, which is a kind of extracellular fluid transport and probably the closest thing to a sewer system in your brain, though it's not entirely devoted to flushing waste. Many types of glia cells are implicated in multiple neurological diseases, and in these domains, it appears they can be both heroes and villains, though many of these areas are still under investigation. But we'll look at what we understand so far about their involvement in disease states as we go through each type of glial cell. So, where do glial cells live? Well, you'll find them everywhere neurons live. You may remember from last episode that in the brain there are as many glial cells as neurons. I couldn't find an approximate calculation of glial cells in the peripheral nervous system, or anywhere outside of the brain really, but I did find an approximate size of a certain type of glia that insulates the axon of some peripheral neurons called Schwann cells. We'll get into more detail on that type of glia in a moment, but one Schwann cell covers roughly 1 millimeter of axon, which is actually a considerable amount of real estate given the general size of a neuron. Though to be fair, axons in the periphery can be quite long. It is conceivable that you could have one peripheral neuron with hundreds of Schwann cells covering one axon. So multiply that by hundreds of thousands of myelinated peripheral neurons, that's a lot of Schwann cells, at least. But there are other glial cells in the periphery, so that's not super great context in regards to the number of glial cells in total. But I hope it helps a little. Let's start with central nervous system glia. In the brain and spinal cord, we have astrocytes, microglia, oligodendrocytes, and ependymal cells. In the brain, astrocytes are best friends with the blood-brain barrier, so we'll get to know that a little better. The blood-brain barrier comes up a lot in health and wellness discussions, especially when we get into supplements and skincare products, including incredibly divisive topics like sunscreens, of all things. So understanding it and its relationship with astrocytes may be of some help to you. Okay, so astrocytes. There's a lot going on with these beauties. Outside of the glial cells that make myelin, astrocytes are one of the most studied of all glial cells. When I did a general search for academic papers on astrocytes, nearly 1 million results popped up. Needless to say, I have not read them all, so much of what I'm about to tell you about them could change or evolve as we keep learning more about them. I mean, that's true for just about everything you'll learn in this series and science in general. But I feel like it's important to be clear that our understanding of glial cells, including astrocytes, is still in development. You may have heard of astrocytes before if you've heard of Marion Diamond. Dr. Diamond was a firecracker of a scientist, researcher, mentor, and teacher, and if you don't know who she is, please look her up. I'll post a link to a little documentary about her. Anyway, Dr. Diamond was among the first generation of neuroscientists and made important discoveries that advanced our understanding of neurons and neuroplasticity and led to even more enormous advancements in our understanding of the importance of enrichment on brain development. But something that she appears to be most noted for is that she studied Albert Einstein's brain. And what she and her colleagues found was that he had more astrocytes in his brain than the average human brain. There are many hypotheses as to why this would be, but Dr. Diamond's lab thought this was because the neurons of a genius would have higher metabolic demands because of all the thinking and problem solving. His little gray cells would be very hungry, and one of the many jobs of glial cells, astrocytes included, is to supply neurons with the ingredients needed to produce ATP, their number one source of energy. This will get clearer as we get through this episode, but do look up Marion Diamond. Dr. Wendy Suzuki, who is a brilliant neuroscientist in her own right, was a student of Dr. Diamond, and in interviews has praised her mentorship and masterful teaching style. But I think one of the sweetest tributes she makes to Dr. Diamond is when she said in her TED Talk that the day she saw Dr. Diamond pull a real human brain out of a hat box and unveiled it to her class, that was the day she knew she wanted to become a neuroscientist. I mean, that's a pretty awesome moment. Okay, back to astrocytes. It should be said from the get-go that there is a great deal of diversity in form and function when it comes to astrocytes. Where they are located in the central nervous system dictates the division of labor they are tasked with, and their form of morphology is somewhat dependent on that as well. But let's begin with a general overview of their anatomy. They are called astrocytes, or sometimes astroglia, because astro is Greek for star, and astrocytes have loads of extensions off their cell body, or soma, that give them the appearance of stars. Those extensions are not dendrites or axons. They're called processes, and at the end of each process is an end foot, which is like a terminal button or end bulb, and it synapses to neurons, as well as to vascular capillaries that make up the blood-brain barrier. And not just one neuron, one astrocyte has so many processes it can synapse with hundreds or thousands of neurons. Like the neuron, they have a soma with typical nucleus and organelles, though perhaps not as many as the neuron. They may have some additions based on the type of astrocyte, and we'll get into those in a minute. For instance, they tend to have glycogen granules, which store glucose molecules. This will make sense when we get into some of its functions. From this general structure, astrocyte morphology has some variation depending on its type. There are a few types of astrocytes present in the human brain. The two main types I'll go over are protoplasmic and fibrous. But another type of note is the interlaminar astrocyte. Its specific function is still not fully understood, but it is abundant in the top cortical layer of the brain. But let's look at the other two because we know a little bit more about them. Protoplasmic astrocytes are found in the gray matter of the brain and spinal cord. If you remember from the previous episodes, your gray matter is the concentration of neuronal cell bodies, and those are found in the outermost layer of the brain, which is also called the cortex, as well as some intersections of the brain, and those areas are generally referred to as nuclei. Gray matter also runs down the center of the spinal cord, and again, that is referring to concentrations of neuronal cell bodies. So protoplasmic astrocytes are most abundant in all of those gray matter areas, and they are tightly integrated into those neuronal concentrations. One of their functions is to make contact with blood vessels in its process end feet, and that particular contact is called perivascular end feet. These contacts help maintain the structure of the capillaries while also drawing nutrients from them to feed neurons. Fibrous astrocytes are found in white matter, which is all of those myelinated axon fibers in the brain and optic nerve and spinal cord. They are organized between rows of axons and project very long processes compared to protoplasmic astrocytes, but have fewer very fine processes. And those also make perivascular end foot connections around blood vessels, but they also contact axonal nodes of Ranvier. If you remember, those nodes are the tiny bits of naked axon exposed between the myelinated oligodendrocytes with scroll hands. You'll need to see the previous episode to really understand that. Functionally, astrocytes have numerous roles. There's a strong emphasis on their heterogeneity in the literature, so I'll give you a general idea of their many functions with the caveat and gentle reminder that some of these functions can depend on where in the central nervous system they are, And these cells are still under investigation and we don't fully understand everything about them yet. But one of the primary functions of astrocytes involves a symbiotic relationship with the blood-brain barrier. The blood-brain barrier is a term that gets bandied about in health and wellness discussions, especially pertaining to supplements, pharmaceuticals, even skincare. So I think it's worthwhile to take a minute to describe its physiology and function, because what it sounds like it is and what it actually is may be a little different than what you're expecting. I myself am a highly visual person. I think visually. So when I first heard blood-brain barrier, I had imagined the arterial version of the Great Wall of China encircling my brain with beefy molecular warriors standing guard, deciding what can enter my brain space. It turns out that's not quite what it is at all. It's actually most of the microvasculature in your central nervous system, and it has a specific cellular and molecular phenotype. So it's loads of very small blood vessels branching throughout your brain and spinal cord that have very specific components in and around its walls that keep it tightly regulated, meaning its structure has multiple built-in safeguards to ensure every ion, molecule, and cell that moves from the blood into the central nervous system is allowed to pass into those vulnerable spaces. The highly restrictive nature of this microvasculature only allows water, oxygen, glucose, and other very small safe substances to cross the barrier into the brain. This protects the brain from toxins, pathogens, inflammation, disease, and so on. This is incredibly important to maintaining homeostasis in the central nervous system. In fact, it's so difficult to penetrate this barrier that it takes an incredible amount of time, effort, and trial on the part of therapeutic drug developers to get drug molecules past it and delivered to their targets in the central nervous system. There are very few small areas of the brain where the vasculature is not as restrictive as the blood-brain barrier, and that's because those areas need to allow hormones to freely cross into the brain. Now, the blood-brain barrier isn't immune to degradation, and some of the culprits that contribute to its failure, or more accurately contribute to the loss of some of its specialized components that keep it robust, are neurodegenerative diseases, stroke, multiple sclerosis, and traumatic brain injury, among others. And to add insult to injury, the dysfunction of the blood-brain barrier during these disease or injury states contributes to the progression of these states. It can also lead to central nervous system dysregulation, which throws off homeostasis, and that could also lead to eventual neuronal degeneration. The three major components that make up the blood-brain barrier are the endothelial cells that make up the vascular wall, parasites that wrap around the endothelial wall, and astrocyte end feet that wrap around the other two components. And within all of these components are specific transporters and ion channels and pumps, and among other functions, they act as added safeguards on top of the already restrictive permeability of the vessels themselves. So if I haven't articulated it well enough yet, unless your brain is massively compromised due to neurodegenerative diseases or traumatic brain injury, including stroke, it's very difficult for foreign substances to cross that barrier. Now, not impossible, but very, very difficult. The astrocytes' role in helping to regulate the blood-brain barrier is several-fold. They are integral to the development of the vasculature's robustness during angiogenesis, including inducing the growth of the cells that make up the vessel wall, secreting all kinds of business that contribute to the growth of the vessels and the formation of strong, tight junctions between the vessel cells that diminishes any permeability. It is believed that astrocytes continue to maintain those tight junctions throughout one's lifetime, which implies that astrocytes are partially responsible for maintaining the structure and restrictive permeability of the blood-brain barrier. Astrocytes also draw nutrients from the blood-brain barrier through their end feet, and they break them down and send components as ingredients to neurons so they can make their own energy. These nutrients are mainly glucose and water. That's why astrocytes have glycogen granules. They store glucose as glycogen. They then synthesize the glycogen and break it down into its substrates, one of which is lactate. The lactate is gifted to the neuron, which it uses to make ATP. And as you all know by now, ATP is the fuel neurons need to do their business. And lastly, as it relates to the blood-brain barrier, an incredibly underrated function of astrocytes is its role in flushing excess waste from your brain. In the periphery, there is the lymphatic system, and that flushes waste from your body. In the central nervous system, there is the glymphatic system that flushes waste from your brain. It can also distribute non-waste molecules to other areas of the brain. This system is formed in spaces between astrocytic end feet and the vascular walls. Think sewage tunnel, but with a capillary running through the middle of it. This space between end feet and blood vessel is filled with fluid and clears out accumulated brain waste and discharges it into the cervical lymph system, which is in the neck area and part of the greater lymphatic system. Brain waste can be anything from excess neurotransmitters to neurotoxins, like possibly beta amyloid, which is the main component of a kind of plaque that is implicated in the development of Alzheimer's disease. The most intriguing aspect of lymphatic flushing is that it happens while you're asleep. It's not active during awake states. So if anyone ever asks you, why do humans need to sleep, or why would we ever put ourselves in such a vulnerable position as sleep, you can tell them. At least one reason is so our brains can flush after a day full of dumping waste. Astrocytes also have loads of functions beyond blood-brain barrier business, one of which is involved with communicating with each other and with other neurons, not by electrical signals, but by chemical signals. Their chemical of choice is calcium, and when the astrocyte receives calcium signaling, it can be a local signal, or it can send waves of calcium signals through its network of astrocytes. Astrocytes also release nearly 200 different kinds of molecules, ranging from neurotransmitters to hormones to growth factors to neuromodulators. It's possible these calcium waves of communication trigger the release of some of these molecules that then communicate with neurons at multiple targets, helping to regulate multiple brain functions, including synaptic neurotransmission, plasticity, and even metabolism. Astrocytes are also involved in maintaining balanced ion concentrations in the brain. They have a variety of ion channels, including my favorite, passive potassium channels, because potassium still likes to wander, and in the astrocyte's case, potassium's Kerouacian ways is what keeps the astrocyte's membrane potential stable and balanced. Astrocytes also have sodium-potassium ion pumps, and that pump contributes to maintaining homeostasis by helping to regulate sodium and potassium ion concentrations all over the brain. The other ion channels, it is believed, contribute to allowing enough electrophysiological flexibility to keep the astrocyte adaptive to its environment. Astrocytes are also like first responders when brain tissue is injured. They're involved in scarring and repair in a process called reactive astrogliosis. Again, they respond in different ways depending where in the central nervous system the injury is and what type of injury it is. But generally, in scar formation, reactive astrocytes rapidly respond to injury by proliferating or multiplying and densely surrounding the injured area. This forms a border or scar around the area and helps protect the injured area while it repairs, keeping inflammatory cells contained and preventing them from spreading into healthy tissue. In terms of neurorepair, astrocytes initiate numerous signaling pathways that trigger a number of responses that help with regenerative repair, including the inflammatory response and the release of neuroprotective factors. Astrocytes also have loads of receptors, and those monitor synaptic transmission of neurotransmitters and neuromodulators. This helps to stabilize neuronal synapses. So in the last episode, when we looked at synapses of the neuron, we just looked at the neuron, but let's add the astrocyte to this all-important junction. So remember, with the neuron, we have the presynaptic button, the synaptic cleft, and the postsynaptic dendrite. Well, the process end foot of an astrocyte hugs the neuronal synapse. The receptors in that end foot are monitoring synaptic functions, and the astrocyte will release proteins to regulate that activity. For example, if you remember, glutamate is an excitatory neurotransmitter, and too much of it left in the synaptic cleft can cause excitotoxicity. Well, astrocytes have glutamate transporters that lend a hand and hoover up extra glutamate from the cleft and break it down into its component parts and give it back to the presynaptic button in a form that can be recycled and turned back into glutamate. Astrocytes also play a key role in the developing brain. This discovery was the work of Ben Barres and his colleagues, and I'll properly introduce you to him a little later on in this episode. His was one of the landmark discoveries that astrocytes have greater purpose than originally thought. Through his and others' work, it was discovered that astrocytes control the birth, growth, and extinction of synapses that help neurons mature properly and properly organize connections with one another, and as mentioned, then monitor these connections to ensure effective communication with each other. He and members of his lab also discovered that reactive astrocytes can have a dark side. This is still under investigation, but in preliminary and ongoing studies, depending on the type of injury, reactive astrocytes would either induce neuroprotective factors that promoted survival and repair, or they induced a signaling cascade that led to the death of the injured neurons instead of repair. This has some pretty strong implications for neurodegenerative diseases and brain injury. So let's recap. What we can extract from all these known and partially understood functions of astrocytes is that they are fundamentally crucial to the maintenance of homeostasis in the central nervous system. From the very teeny tiny atomic levels all the way to the entire nervous system, they maintain and modulate the blood-brain barrier, protecting the central nervous system from invasive pathogens and toxins, as well as draw in glucose from the bloodstream and break it down and give its components to neurons to make ATP. They also keep ion concentrations balanced in the brain. They regulate molecular stability by monitoring and supporting neuronal and synaptic connectivity and neurotransmission. They repair injured central nervous system tissue. They ensure proper synaptic birth, development, and pruning. And they maintain the glymphatic system for the entire brain as an organ. And all of these things are necessary for central nervous system homeostasis. Okay, next up are microglia. Microglia are another type of glial cell with functions that help maintain homeostasis in the central nervous system. Like other glial cells, their functions are still under investigation, but what we understand of them so far is that their main gig is sweeping your brain and spinal cord for dead cell bits or debris and snacking on them. They also have an incredibly important role in the immune response, but more on that in a minute. Let's first look at their structure. They are small, micro means small. They are the smallest of the glial cells. Their morphology takes on two forms. When they're inactive and resting, they are a little similar to astrocytes in that they have long, fine processes that branch out from their soma. When they are active, they sort of scrunch in those processes close to the cell body, and the cell body swells, so they end up looking more like an amoeba than an astrocyte. Now, resting and inactive are really colossal misnomers because in these states, microglia are neither really resting or inactive. They're actually quite dynamic. They're moving all around the central nervous system using their processes to sweep or survey their local environment, looking for things to clean up. So some researchers prefer to call the inactive state, more accurately, the surveying state. As far as function is concerned, we are learning that these dynamic little beasts have more roles than originally believed. What we're pretty certain of is that during their surveying state, they are constantly moving about the interstitial fluid, which is the extracellular brain juice between neurons and glia. So they're searching the interstitial fluid spaces for contaminants to clean up, and that can be myelin debris, dead or apoptotic cell bits, microbes, protein corpses, and the like. When they find these bits, they sort of grip the bits with the end bulbs of their processes and pull it in, engulf it, and then digest it, which makes them sound a bit like something that escaped from a Guillermo del Toro film. But not to worry, this is just regular old phagocytosis. We've come across this word a few times before if you followed this series from the start. But phagocytosis is when certain cells engulf or ingest waste floating around the intra and extracellular spaces, and they digest it by unleashing organelles like lysosomes, which use enzymes to break down whatever was ingested. The microglial find, eat, digest mechanism occurs while in the surveillance state. When injury or disease or infection invade the central nervous system, signals alert microglia and they kick into activate mode. And as mentioned, when they activate, they shapeshift into something more amoeba-like, and they multiply and get a move on to the injured or diseased site to take care of business protecting, cleaning, and organizing. And that can be anything from eating up the debris from the injury or bacteria from the infection to releasing its own signals to initiate the inflammatory response and or recruit the assistance of other immune cells in order to restore the tissue back to homeostasis. However, like responses elsewhere in the body, persistent or chronic inflammation can create more jobs than microglia can handle, which can compromise their neuroprotective abilities and leave neurons vulnerable to degradation. Microglia also appear to have significant roles during brain development. During fetal development, they could be assisting in neurogenesis by phagocytosing neurons in extraneous synapses. This is totally normal. Prenatally, babies produce twice as many neurons as an adult, so microglia are possibly monitoring for way overproduction. Microglia may also play a role in neuronal differentiation, which is making sure the neuron makes it to its final destination in the nervous system. After birth, microglia may be involved in myelin development and regulating synaptic development, growth, and plasticity, and quite possibly synaptic pruning. And that is also a normal part of development, because remember, we're born with way more neurons and synaptic connections than we need in adulthood. And the pruning process happens at certain stages of development where some of these neurons and connections that aren't being used are phagocytosed. Think of pruning a tree for the health of the tree. Pruning definitely happens in the developing brain, and it's quite possible that microglia could be involved in that process. Okay, up next are oligodendrocytes. And you may remember these special cells from the last episode when we talked about saltatory conduction of action potentials, which are electrical pulses that fire down the axon during neurotransmission. Saltatory conduction is when the electrical signal leaps over the myelin sheaths to each node in between the sheaths, which greatly speeds up the signal transmission process. While oligodendrocytes, or oligodendroglia, are the myelin producers for axons that are myelinated in the central nervous system. Like most of the other glial cells, we're still learning about the functions of oligodendrocytes outside of myelination. Even within the bounds of myelination, there are still some things that aren't fully understood yet. So again, I can tell you what we're pretty sure we understand so far, but this information could change or evolve as we learn more about them. So first, their structure is intriguing. It's a bit like the other glial cells we've discussed so far in that they have a cell body, or soma, and many processes that extend from the soma. Except if you remember from the last episode, oligodendrocytes have at the end of their processes these sort of Swiss roll hands called myelin sheath. Well, they don't start out as Swiss rolls, but as these cells develop and find their axons and extend their processes toward them, the ends make contact and begin to wrap around the axon in a dual direction, meaning each layer rolls under the next and sort of widens as it wraps. Each myelin sheath can wrap up to 100 layers, and their length appears to vary depending on the size of the axon and where in the central nervous system it is. But some numbers that came up are somewhere between 10 and 12 nanometers in length, which is very, very tiny. There are 1 million nanometers in a millimeter. But another source said that they can be up to 1 millimeter in length, so not exactly sure on that one. The myelin sheath Swiss roll hands are mostly made of lipid membrane, and the layers are quite compacted, and that compaction leaves the myelin membrane inhospitable to just about anything else, except for a couple of proteins, a smidge of cholesterol, and a bit of water that rounds out its structural matrix. But this matrix and its compaction make it highly stable, and it has to be considering that it endures so much electrical conduction. There is a wee space between the innermost myelin sheath Swiss roll layer and the axon itself that isn't compacted myelin, and that's called the periaxonal space. And this is a rather interesting space because that innermost layer of myelin sheath is able to house transporters that shuttle blood-derived glucose components into that periaxonal space, and it's picked up by the axon transporters and converted into ATP by one of the bazillion mitochondria inside the axon. So not only do oligodendrocytes insulate the axon and speed up signal conduction, they also appear to support neurons metabolically. How they source the glucose or its components to begin with could be drawing it in from extracellular spaces through transporters or indirectly through contact with astrocytes. But how it finds its way through the myelin sheath to the axon is a bit of a mystery, since the compaction of that myelin doesn't seem to be conducive to the movement of anything. But one paper suggested that there are a bunch of strategically located cytoplasmic pockets throughout the layers of myelin that enable the transport of these metabolites through 100 layers of tightly compacted myelin to the transporters in the innermost layer of the myelin sheath. So among their many other roles, oligodendrocytes could be snack mules for neurons. And if you're a parent, you'll totally relate. Anyway, it is somewhat comforting to know that oligodendrocytes help carry the metabolic load, because if you remember from the last episode, and I've mentioned it multiple times in this episode, one resting neuron can burn billions of ATP molecules per second, and neurons do not store glucose, so they rely heavily on support glia to feed them. Okay, back to oligodendrocyte neuroprotective and metabolic roles. Given that these are pretty crucial to central nervous system homeostasis, when oligodendrocytes malfunction, it's not surprising that their demise can lead to neurodegenerative pathologies. I gave an example of oligodendrocyte demyelination of the optic nerve in the previous episode and its implication in the development of glaucoma. But there are other neurodegenerative pathologies that involve oligodendrocytes and their myelin degradation. One that receives much due attention is multiple sclerosis, or MS. MS is both an inflammatory and neurodegenerative disease characterized partially by myelin loss from central nervous system neurons. But the etiology of MS is still not super clear. Myelin degradation leaves axons exposed, which would compromise their efficiency. It would also leave them somewhat unsupported metabolically, and these together could lead to neuronal degeneration. In circumstances where neurons and their axons get injured and demyelination occurs, among what generally follows is the proliferation of more oligodendrocytes and the prompt remyelination of the injured axons. This requires oligodendrocyte precursor cells maturing into full-grown proper oligodendrocytes, with their fully functional myelinating processes wrapping those Swiss roll myelin sheaths around the naked axons before they atrophy. Except within MS-affected areas, which might also be referred to as an MS lesion, in those areas where demyelination has occurred, there may be some factors that prevent the precursor cells from maturing properly, and so remyelination can't happen, or at least not sufficiently, leaving the injured axons to deteriorate. You might be wondering where the other superhero glial cells are, and why they aren't kicking in to save the day. Well, they kind of are, and that could also be part of the problem. They recognize injury and activate the inflammatory response, but for reasons not yet totally understood, that inflammatory response seems to become self-perpetuating in MS, which is not good and creates either a local or global neuroinflammatory environment, and either of those can contribute to the prevention of oligodendrocyte precursor cells becoming full-grown oligodendrocytes. There's a lot more going on in MS pathology than I've said here, but I hope this helps give some context to the importance of oligodendrocytes and what can happen to them in neurodegenerative disease states. I've posted a paper in the show notes that reviews MS pathology and the roles of glial cells and oligodendrocyte precursor cells in its progression. It's open access, meaning it should be available to anyone. I try to include as many open access papers as possible in the show notes, but that paper may be of some help to you, especially if any of you are in close proximity to someone who has MS. The next central nervous glial cells we'll look at are ependymal cells, and they line cavities where there is cerebral spinal fluid. In the brain, they line 4 ventricles, and in the spinal cord, they line the central canal. One of their main functions is to help produce, secrete, and maintain cerebrospinal fluid in these cavities. Their structure is a little different than the other glial cells in the central nervous system. Their soma is kind of cubic, and instead of processes, they have cilia, which are hair-like fibers that oscillate. Their cilia project into the ventricle and canal cavities to keep the cerebrospinal fluid moving and sort of wafting nutrients and other substances toward the neurons. The cilia also filter out any substances that shouldn't be floating around in the cerebrospinal fluid. So how do ependymal cells help produce cerebrospinal fluid? Well, there is a layer of these cells surrounding blood vessels in the choroid plexus, which lines the ventricles in the brain. Ependymal cells draw water and other molecules such as ions, oxygen, and growth factors from blood into the cells through uptake transporters. The substances make their way across the ependymal cells and are secreted into the lateral ventricles to form the cerebral spinal fluid. The ependymal cells that surround the choroid plexus are connected by tight junctions, much like the cells that line the blood-brain barrier, and for similar reasons. This forms a blood cerebrospinal fluid barrier, which prevents substances and fluids leaking into and out of the blood vessels of the choroid plexus. This, of course, prevents unwanted substances from getting into the cerebrospinal fluid and circulating through the ventricles and into the rest of the central nervous system. So the last group of central nervous system glial cells I will briefly discuss are radial glia. Radial glia are active during embryonic development and help with the genesis and differentiation of neurons and other glial cells. I think I want to save the bulk of information about developmental radial glia function for the next episode on neural development. But I do want to mention that there are also radial glia-like cells in adult vertebrates and invertebrates with a very similar phenotype to embryonic radial glia. So I'll go over some aspects of this phenotype now, but we'll really get into function in the future when we can spend more time on them because they are really special cells and they are intimately involved in neuro and glial genesis. Neurogenesis is the birth and differentiation of neurons, and in humans, neurogenesis occurs in stages up until adulthood. After the human brain is fully formed, neurogenesis, particularly in a brain area called the hippocampus, is a very prickly subject that deserves way more context than what would be appropriate to delve into in this episode. So I'll stick to structure and hint at function for now. Okay, so embryonic radial glial cells have a soma with a typical nucleus and organelles, and usually one very long process radiating from it. In embryonic development, that long process provides a scaffold for baby neurons to use as a guide to find their way to wherever they're meant to go and live until they are pruned or die on their own. Embryonic radial glia are also progenitor cells, which means they themselves can develop into specialized cells like neurons or glia. And radial glia-like cells in adult vertebrates have similar structure. So somas and long radiating processes, and some of the areas they live are the spinal cord, retina, cerebellum, hippocampus, the lining of the brain ventricles, and possibly the olfactory bulb. Beyond morphological or structural similarities, both embryonic and adult radial glia have similar gene expression. This morphology and marker expression is what rounds out their phenotype. I want to be very clear that most of what we understand about radial glia-like cells in adult vertebrates have been studied in animal models or in a dish. Okay, that was glia of the central nervous system. Now we'll take a peek at the glia of the peripheral nervous system. A quick reminder that your peripheral nervous system refers to all the neurons and their axons in the periphery that either sense things, so sensory neurons, which send signals to the brain and spinal cord to figure out what action needs to take place, or motor neurons that receive signals from the brain and spinal cord to innervate muscles and generate movement. The first glial cells we'll look at in the periphery are Schwann cells, which we briefly mentioned in the previous episode and earlier in this episode. Schwann cells are like oligodendrocytes in that they can be myelinating cells that wrap around the axons, but these are just for neurons in the peripheral nervous system. Both motor and sensory neurons can be myelinated with Schwann cells. What makes them different from oligodendrocytes is that one Schwann cell wraps its whole darn self around one part of one axon. It doesn't have a bunch of arms with Swiss roll hands reaching out to multiple axons. Its whole self is the Swiss roll, and multiple Schwann cell Swiss rolls line the peripheral nerve or axon. There are also non-myelinating Schwann cells, but we're only going to look at the myelinating kind in this episode. So let's look a little closer at the structure of these myelinating beauties. The Schwann cell starts out like a regular blobby cell with a nucleus. It's tightly covered in something called basal lamina, which is a layer of extracellular proteins, collagen, growth factors, and other molecules that are used to receive and send signals for all kinds of business, including when and where to initiate myelination and on which axons. I like to imagine the basal lamina is the Schwann cell's supersuit. The actual process of Schwann cell myelination is a modern love story befitting of multiple genres. Here's what happens. An axon sends a "hold me, Schwann" signal from areas all along the axon to Schwann cell blobs, and since the Schwann cell blobs would love nothing more than to canoodle the part of the axon that called them, the sweet blobby cells wearing their basal lamina supersuits find their way to their axon fates. That is for real what this dynamic is called. The area of the axon the Schwann cell responds to is termed the myelinating fate. And I can't think of this process without imagining the Schwann cell reaching the axon and saying in its blobby code, "You are my fate, you are my density," in an adorably George McFly way. And so then Schwann begins to wrap itself around that part of the axon like a blankety hug, and then keeps wrapping each layer underneath the previous layer and sometimes up to 300 layers of love. And like oligodendrocytes, those myelinated Schwann cell layers are tightly compacted and supports that part of the axon structurally and metabolically, and speeds up electrical saltatory conduction velocity. But unlike oligodendrocytes, if that axon it's hugging gets damaged, Schwann cells will call in their macrophage friends to help them cannibalize the dead axon and myelin bits. Okay, so that's maybe where this love story gets a bit weird, but hold your matrix because it writes itself. After it helps digest the dead parts of the axon and any myelin debris, the Schwann cells initiate repair mode. And what happens now is the basal lamina supersuit morphs into a tube, and the Schwann cells that weren't injured and phagocytosed shapeshift into long, tube-like structures and arrange themselves in a cylindrical formation called a Buhner band all along the inside of the basal lamina supersuit tube. And this repair mode Schwann cell phenotype forms all the way from the axon stump to its former intended target. This creates an environment conducive to facilitating the regrowth of the injured axon. The axon will sprout new growth fibers, and those fibers are guided by the Schwann cells to make their way through the Buhner band, and while it guides the axon through its scaffolding, it secretes growth factors and collagen and other molecules from the basal lamina supersuit tube that aid in axonal regeneration. Schwann cells are maybe the supportiest of all the nervous system support cells. They are the cellular version of that dream partner or friend who, when you're sick, tucks you in and says, "Darling, I got you. You heal. I'll do the dishes. I'll vacuum. I'll make you bone broth. You just rest." And regenerate in this case. And it doesn't end there. When regeneration is complete, the Schwann cells morph back into their myelinating cell phenotype and get their cuddle back on. This process doesn't happen in the central nervous system. This is a massive difference between Schwann cells and oligodendrocytes. Important note, however, depending on the size of the axon, it could take months to fully regenerate. And in adult humans, even our supersuit Schwann cells can exhaust their resources and lose their ability to continuously care for the axon for months and months through to complete regeneration. I believe Schwann cell longevity for lengthy repair processes is a target for therapeutic drugs, but I'm not sure how close we are to an effective product. Okay, the next peripheral nervous system glial cells we'll look at are satellite glia. There isn't a ton of information on satellite glia, but the emerging literature shows that, like many specialized cells in the nervous system, their functions may be dependent on where they are located. They are unlike any of the other glial cells structurally, but functionally they share some similarities to astrocytes. But let's take a look at their structure first. Satellite glia are not at all interested in respecting personal space. They wrap around the somas of groups of neurons that live in clusters called ganglia in the somatic and autonomic nervous systems of the periphery. And if you remember, the autonomic nervous system is subdivided into the sympathetic and parasympathetic nervous systems. And I say wrap, but it kind of looks more like these neurons slapped a bunch of flat fried eggs with arms called perineuronal processes all over their somas. Satellite glia have a large nucleus and the usual organelles, and as I said, they envelop the exterior of the neuron soma in somatic and autonomic ganglia. Not just one satellite glial cell, but multiples, until the soma is surrounded. Their membranes have receptors and ion channels that are in close communication with the neurons on which they inhabit. On sympathetic neurons, satellite glia cell processes will cover synapses and envelop axons and dendrites as well. In some sensory ganglia, groups of 2 or 3 neurons might share a common satellite glia envelope. Functionally, they are similar to astrocytes in that they might help neurons maintain homeostasis by assisting in the regulation of their metabolism, synaptic function, and structure. In addition to those functions, they appear to express genes that are involved in the synthesis and metabolism of lipids and cholesterol, which may be where neurons source their lipid uptake because neurons don't seem to do it themselves, or at least not efficiently. There is also some evidence that satellite glia may contribute to chronic pain processing in sensory neurons. Also of note in the health and wellness spheres, and somewhat controversial, is that there is some research that suggests satellite glia may prevent the resurgence and spread of the herpes simplex virus, which apparently lays dormant in sensory ganglia after the primary infection. It's possible they may even rid sensory ganglia of the virus altogether and help to repair the nervous system when it's past its latency stage. Why this is controversial is because there doesn't seem to be a clear way to fully demonstrate and validate satellite glia response to the virus with current study models. Though that could change, but rightfully so, some researchers are reticent to say with any certainty what kind of involvement satellite glia have on latent viruses. Why does this matter? Well, if we can improve the methods by which we study satellite glial response to virus infections, that could inform treatment targets for the herpes virus in particular. It could also provide a springboard to study satellite glial response to other even more harmful latent viruses like HIV, or non-latent viruses, even various iterations of the coronavirus. So it's probably worth finding models that work. Okay, last but never least are glia of the enteric nervous system. A quick reminder that the enteric nervous system is a network of neurons in the walls of the gastrointestinal tract. It receives input from and sends output to the sympathetic and parasympathetic nervous systems, but it's mainly self-contained. It reflexively activates digestive functions that involve peristalsis, which is the contraction and relaxation of the digestive tract. It's also involved in the secretion of digestive fluids and absorption of nutrients into the digestive tract and beyond. What we understand of enteric glia is still emerging in the literature, and much of what we do know has been studied only in animal models. Their structure is somewhat like the astrocyte in that they have a soma and lots of processes, and they live in the intestinal wall with enteric neurons. They appear to behave like other glia in that they participate in regulating homeostasis by supporting neurotransmission, regulating inflammation, and protecting against invasive pathogens. On top of that, and more specifically to gastrointestinal function, enteric glia help regulate intestinal reflexes like gut motility, which is the process of moving food through your body, and in particular, peristalsis, which is one component of gut motility. Something you may have heard bandied about in health and wellness circles is intestinal hyperpermeability, or leaky gut. If you haven't heard of it, it refers to when the protective integrity of the intestinal barrier is compromised to the extent that it leaves your gut vulnerable to absorbing more than just water and nutrients, like bacteria or pathogens, which could eventually end up circulating in the bloodstream. What the research suggests is that enteric glia have an important role to play in intestinal barrier function, but we're not yet certain what their function is in healing a compromised intestinal barrier like that of leaky gut. As efforts to understand enteric glia advance, hopefully it will inform new ways to treat and protect gastrointestinal health. We will spend more time on the enteric nervous system in the future episode, and there we'll look more closely at the gut-brain axis, what the research says about intestinal hyperpermeability or leaky gut, and how and why the gut produces 95% of your serotonin. Okay, folks, that's a wrap on glia. We've come to maybe my favorite part of these episodes, and that's when I get to introduce you to some amazing scientists who have been or are in the trenches investigating and expanding our understanding of the extraordinary ways our bodies function. Though there are many incredible scientists researching glia, for this episode, I want to focus on one scientist in particular because there is so much to tell you about him. I mentioned him briefly when discussing astrocytes earlier in this episode. His name is Dr. Ben Barres, and he was chair of the Department of Neurology and a professor of neurobiology, developmental biology, and neurology at Stanford University. And as I said earlier, he was one of the world's leading glial cell researchers. He passed away in 2017 from pancreatic cancer at the all too young age of 63. His contributions to the advancement of our understanding of glial cells and neuroscience as a whole are just part of his legacy. He was the consummate scientist, asking the big questions no one else wanted to ask or answer, and he persevered until he had answers. When his other colleagues were chasing the sexy neuron, Dr. Barres was asking, yeah, but could the neuron even be sexy without glia? And if you've made it this far into this episode, you know the answer is a resounding heck no. Just some of his contributions include generating tools that made studying glia and their functions even possible, and then generously making those tools widely available to the scientific community, which is atypical in what appears to be a somewhat territorial profession. Dr. Barres's dedication to unraveling the mysteries of glia led him and his colleagues to discover that glial cells weren't just passive brain guts holding neurons in place. But as mentioned earlier, astrocytes in particular help with the formation, activation, and extinction of synapses, as well as help neurons mature properly and keep their synaptic connections properly organized and functional. He and his lab also found that astrocytes work with glia to prune excess synaptic connections during fetal development. And also mentioned earlier, he and his lab discovered that astrocytes in their reactive states may either induce the release of neuroprotective factors that promote survival and repair, or they induce a signaling cascade that leads to the death of the injured neurons instead of repair, depending on the circumstances. These are just some of the landmark discoveries he and his lab have made. There are a number of other glial cell contributions, as well as many other contributions to the field of neuroscience. Contributions that garnered him multiple prestigious awards. I'll post links to a few papers in the show notes below that describe the depth and breadth of his contributions. But as I said, that's just part of his legacy. When I started to collect articles about Dr. Barres's life that were written by those who knew him, there wasn't one paper that didn't mention what an extraordinary human he was to anyone and everyone whom he met. He was beloved by his students, colleagues, mentors, friends, and even students he didn't officially mentor. but for whom he found time to support academically. Many students outside of his lab considered him a surrogate mentor. It seems as much as he gave of his time and resources to research, he poured himself into mentoring his students and preparing them to lead their own labs. When one of his academic progeny, Dr. Beth Stevens, left his lab to take a position at Harvard University, she said, and I quote, "Ben told me, 'Take this work with you to your new lab, Beth. Nobody can do it better than you.'" She continues, quote, "Mentors aren't always so generous about ceding areas of research initiated in their lab to trainees headed elsewhere. But Ben was a very special person. Not only was he an incredible scientist, but he also cared deeply about other people, especially his trainees. We were his kids." End quote. When Dr. Barris was given his terminal diagnosis, he set to work ensuring every one of his trainees was set up for mentorship and funding so they could see their research goals through to completion. At the end of his days, he was confined to hospice care in his home, and even then he updated reference letters for all of his previous and current trainees and made sure they'd be available to them after he was gone. For most humans, we'd be exceedingly fortunate to be able to leave a legacy even remotely close to that honorable. But Dr. Barris's legacy doesn't end there. He was also a highly vocal advocate for the fair and inclusive treatment of women and other underrepresented communities in scientific fields and academia in general. When Dr. Barris gave lectures and presentations about research from his own lab, including his very last public presentation, he'd interrupt himself midway through the presentation to raise awareness about sexual harassment in academia and suggested guidelines that would protect junior female scientists. If he came across committees related to funding and academic recruiting or scheduled meetings with panels that were primarily white and male, he'd advocate for the inclusion of women and other underrepresented communities. He'd even preemptively contact science event organizers to ensure any moderating panels were sufficiently inclusive and ensure the organizers had appropriate sexual harassment guidelines in place to protect students and junior scientists. And if people didn't listen to him, he would relentlessly advocate until there was visible change. There's more. Probably the most beautiful and harrowing aspect of his life and legacy was Dr. Barris's unending supply of perseverance and resilience, also known as grit. At the end of his life, he said he had amassed grit in spades, and he believed that this interminable supply of grit developed out of a need to navigate decades of emotional pain, confusion, and despair that came from living in a body that was never his. Dr. Barris survived the first 44 years of his life with undiagnosed and untreated gender dysphoria. Gender dysphoria is the severely felt incongruence and dissonance one can feel when their internal perception of their gender identity doesn't align with the outward anatomical representation of self. There are many possible physiological pathways that could lead to gender dysphoria. Some could emerge as early as fetal development. Ben wasn't certain of his particular pathway, but as a scientist and a voracious researcher, he hypothesized, based on human and animal data available at the time, that fetal exposure to a testosterone-based anti-miscarriage drug that his mom took while pregnant with Ben and his twin may have masculinized his brain development. To understand better what that means, we're going to look at brain development in the next episode, so that might be of interest to you. But I want to be clear, though, that this is not the only pathway that leads to gender dysphoria. Again, there are many, but this is one possibility in Dr. Barris's case. Ben was born anatomically female, but from as early as he can remember, he aligned with typical male personality characteristics, which influenced the way he navigated his world and made it near impossible to fit into peer groups. Growing up, he had a strong proclivity towards toys, clothing, subjects, sports, and peer groups that were socially deemed masculine. He was also born with a reproductive condition called Mullerian agenesis, which prevents the proper development of a uterus and vagina. So though he was born with observable female sex characteristics on the outside, He was missing most of the rest of those reproductive characteristics on the inside. Dr. Barris didn't think this reproductive anomaly contributed to his gender dysphoria, but it did further separate him from peers growing up. For decades, he experienced intense emotional discordance and anguish of being a male trapped in a partially female-presenting body. And he had to manage this long before the medical community had words, let alone diagnoses, for what he was experiencing. He was bullied and ostracized by peers and even family. The dysphoria was deeply confusing and disorienting, and no amount of trying to accept what his external body communicated to the world worked, and every attempt sank him deeper into despair. And he carried that despair while going through years and years of medical and graduate school, honing his research acumen and making prominent discoveries for the advancement of glial research. That's more than an average person could bear. He thought about ending his life numerous times throughout those years because the intensity of the incongruence was too heavy to carry. At 41 years old, when he was diagnosed with breast cancer, he had a double mastectomy. He described it as a huge relief to be rid of this visible representation of someone he wasn't. Not long after his mastectomy, he came across an article about a transgender activist. It was the first time he had heard the word transgender, and the first time he could label what he had been experiencing his entire life. He sought clinical expertise from the gender clinic at Stanford University, where he was officially diagnosed with gender dysphoria, and wanted more than anything to transition from female to male. Nature had already taken care of most of his transition. He had never had a uterus and had already had a mastectomy due to cancer. Hormone treatment was all that was left, and that would induce secondary sex characteristics like facial hair and lowering his voice. But he had only just firmly established his lab at Stanford at that time and was deeply concerned that if he fully transitioned outwardly and socially, he would be rejected by his colleagues, lose funding for his lab, and end his career. A career that was just on the precipice of making those landmark discoveries mentioned earlier. At this juncture, he saw only two options. He could transition or he could end his life. What he couldn't do gonna need a minute. What he couldn't do is carry the anguish of gender dysphoria any longer. After receiving immediate and strong support from his former mentors, he decided to transition and sent a letter to his colleagues and friends asking for their understanding, to which he received an outpouring of support and kindness. Dr. Barris made a courageous choice. If he had chosen the alternative, he'd have been lost to us much sooner than when he was actually taken from us, and we would have been bereft of his incredible contributions to humanity and science. A huge thank you to his colleagues and mentors for being loving and decent humans, because the phase of life that followed that critical juncture was pivotal for Dr. Barris and his contributions to neuroscience, his devotion to mentoring his students, whom he loved like they were his own children, as Dr. Stevens said, and for advocating for the fair and decent treatment of women and other underrepresented groups in science. If ever there was anyone who walked this earth who was the embodiment of grit, it was Dr. Barris. I'm truly honored that I get to share a bit of his story with you. There are many others out there who have done a beautiful job articulating his life's work. The folks at Neurotransmission's YouTube channel did a lovely tribute before he passed away. I believe it's still in their video list. They have a great channel regardless, so you may want to check them out anyway. But I'll post a link to their tribute and to others. Dr. Barris also wrote a memoir shortly before he passed away. It's called The Autobiography of a Transgender Scientist. I believe it's available through MIT Press. It's written in a way that seems very much like how I'd imagine a conversation with Dr. Barris would be like. It feels like you're one of his students, just kicking back with him and others, eating ice cream sandwiches and listening to him regale us with stories about his life and discoveries. There's no formality to it, It's not hyper-academic. It's written the way he speaks. I can hear his voice when I read it and can sense his bubbling exuberance when he gets into all things GLIA related. I can feel his sorrow when he remembers what it was like to live in the wrong body. Conversely, I can sense his utter and total relief when he decided to transition, and I can tell he was quite chuffed and even verklempt by the overwhelming acceptance he received by his colleagues. It's a worthy read, especially if you'd like to know more about his life and, of course, GLIA. And that's all for this episode. I leave you with this quote from Dr. Barris's friend and colleague and MIT Professor Emerita Nancy Hopkins. Quote, "There is something deeply mysterious about Ben that may be impossible to explain. For all his outrage at the world's unfairness, the pain that accompanied his early gender confusion, the risks he took and people he might have offended, he was the most beloved scientist I've known and the most loving. He could charm you with a conspiratorial smile as he contemplated some new unfairness he planned to tackle, or envelop you in love and support when he sensed you were flagging or fearful. One felt safer because he existed." I couldn't agree more. Until next time, lovelies, take care out there.